Hello there, future doctors. Welcome back to yet another video from Best Medicine. Today, we're going to be starting a new chapter called Organization. Organization would stand for how cells make up tissues, and tissues make up different types of organs, and then how organs would give different organ systems, right? Organization. So, what is organization? Organization in multicellular organisms. Remember multicellular organisms like plants and fungi and animals, right? These are large and complex, which means that these are composed of various interconnected systems of organs, right? For example, in humans, we've got the heart, we've got the muscles, we've got the brain, right? We've got the small intestines, the large intestines, all of those. So those organs actually come from groups of cells called tissues. And tissues, of course, would come from cells. So let's begin from the very beginning. Cells. Cells, they serve as fundamental units, right? Cells, we've already studied what they're made up of, how they look like, and how they can become specialized. So cells serve as the fundamental units because these are the smallest living part of an organism that can sustain itself and stay alive, right? Now, cells can become specialized to perform specific functions. For example, muscle cells, sperm cells, root hair cells, xylem cells, right? And the process through which cells develop specialized functions is known as differentiation, which we've already done. Remember, differentiation was the sequential turning on and off of genes to specialized cells. So specialized cells can combine to form different tissues. So if you have one cell, that's just a cell. Let's draw a cell here, and so that's just a single cell. And then if similar cells, groups of similar cells, come in together and start doing a function, that's called a tissue. Okay, and then tissues, multiple tissues can combine to form an organ, for example, the heart. So specialized cells can combine to form tissues, which in turn organize into organs, ultimately contributing to the formation of complex organ systems. Now, large multicellular organisms, such as elephants, they possess intricate internal systems responsible for the exchange and transportation of essential substances and other functions. So, of course, larger multicellular organisms, we have many different types of organ systems. Okay, so coming straight to tissues, remember we've already finished cells in its own chapter, so today we're going to be learning about some types of tissues. And as promised, we're going to begin with the muscle tissue, which I promised to teach you last time when we did cells. So muscle tissue. Of course, we're already aware of what muscles are, right? Muscles make up muscle tissue, which can contract. So muscles have this ability to contract. That's how they help move things, and relax when they're not contracting, right? When they're not using energy, they relax. And what this does is it facilitates movement. So you're able to move things. One example of this would be voluntary movement that you do to move your arms and legs and be able to walk and speak, right? That's voluntary movement, and that is conducted by skeletal muscles. So skeletal muscles help in voluntary movement, such as walking, speaking, movement of head, and things like that. So here's a group of muscle cells making up a muscle tissue. These are long fiber-like materials, and they help form a muscle cell. So for example, if you could have something like a bicep, right? And I think we've already drawn this diagram before, but if this is a hand, right, and then you'll have the bicep here. The function of the bicep, which is a skeletal muscle, is to be able to contract. So if this bicep was to contract inwards, it will shorten. And as it shortens, it will pull the arm towards the shoulder. But if it relaxes, then the arm will drop down, right? Or maybe even the tricep could contract at the same time while the bicep relaxes. So these are skeletal muscles. Next up, we have smooth muscles. So the other kind of movement that we have is involuntary movement, such as moving food along the esophagus, along the stomach, along the small intestines. Also, involuntary muscles of the iris of the eyes, which help us dilate and constrict our pupils. More on that later. And the smooth muscles of the trachea. Trachea is the air pipe that leads to the lungs. So smooth muscles are involuntary muscles that we don't have any control of, and they also help in movement of things. Then thirdly, we have cardiac muscle cells, and therefore cardiac muscle tissue. The cardiac muscles help form the heart, so they help in contraction of the heart, which eventually then pumps blood, okay? So these are the three types of muscles, and muscle tissues. And yeah, so eventually we get a meme here. Okay, this is a funny one. He's probably thinking about other girls, and he's thinking, well, I can't believe you have three types of muscle tissues, right? And hopefully, this is you guys today, because you have learned something fascinating. Okay, so next up we have glandular tissue. Glandular tissue basically means the tissue that is able to secrete hormones or enzymes. So the one that forms glands, right? Do you know any glands? Can you think of any glands? We have many glands in the body, and they are covered in their own chapter called the endocrine system, the hormonal coordination system chapter. But just to mention a few, we have the pituitary gland that is in the brain. We have the thyroid gland that is in the neck. We also have the adrenal glands that secrete adrenaline, right? So we have so many different kinds of glands, and they can secrete hormones and enzymes. So the way this would work is that you would have a cell, you would have a gland. So let's say here's a gland, right? And then this is probably secreting some stuff. So what it really has is that it's got cells here, okay? So that's a cell, and then a cell is making something in vesicles, right? And then let's make this yellow stuff here that is inside this vesicle. This vesicle moves along to the cell membrane and fuses with the cell membrane, releasing the stuff over here, okay? Now, either it can go into a blood vessel. If there's a blood vessel here, this yellow stuff could be a hormone, could be usually if it's a hormone, it will enter the blood. So it will enter the blood and go to distant organs. An example of this could be insulin. If you're making insulin, that's a hormone, it will go into the blood and then travel down to a distant organ like a muscle or liver. Another example of this would be, so this is called endocrine transport. Endocrine, endo means in, so it's inside the blood. The other kind of production of hormones, secretions, would be if a gland is secreting, for example, enzymes. That's exocrine secretion. So if you have a cell, let's say, here's a cell, and then this is just secreting enzymes, but they're just going out and they're not really entering into the blood. So the same thing is going to occur here. You have these vesicles that contain the material that you're secreting, most probably an enzyme, and it's going like this. It could be sweat as well, as sweat glands are also exocrine glands. So it's gonna go ahead and come to the cell membrane and then fuse with the cell membrane, releasing its contents outside. And then they just stay outside and diffuse wherever, right? They don't actually go into the blood and travel to distant organs. And that is called exocrine secretions. Exocrine secretions. Secretions. Examples of exocrine secretions would be sweat. Another example of this would be enzymes in the digestive tract, for example, carbohydrates, amylases, etc. Okay? Endocrine secretion example is insulin. Another example is glucagon, adrenaline, right? And so on. So that's glandular tissue. Okay, here's a meme. When America finds out sebaceous glands produce oil. Sebaceous glands are present in the skin right next to hair follicles, and they secrete an oily substance. You ever heard of people having oily skin? That's because their sebaceous glands are producing oil, okay? Next up, you have epithelial tissue. Epithelial tissue, it's a very easy term to understand. Basically, epithelium is a word that is used a lot in any books of AQA biology, even A-levels biology that you read. Epithelium, essentially, they throw these words to you, but they don't actually explain what this means. So here's the explanation. What does epithelium mean? Epi means on or upon, right? So the word epi means on something, covering, or upon. So if you have an organ and you have a layer of tissue that's covering it, let's say you have an organ, right? And then you have a layer of tissue, cells, right? Groups of cells that is covering it, then that's the epithelium of it, okay? Covering on the outside epithelium. If you have a blood vessel and it's covering on the inside, then that's the internal epithelium, right? That's still the epithelium, but on the inside, okay? So epithelium essentially is just a group of cells that can go like this, right, and surround an organ. Here's an organ, and then it's got this layer of covering on it. So, examples of epithelium would include the intestinal epithelium. Remember the intestinal epithelium from the last video? Today we're going to be doing more details on it. Also, respiratory epithelium, which surrounds the inside of the trachea and the bronchi. Okay, so here's an example of a microscopic slide image of an epithelium. This here is an organ, okay, and covering it is a layer of cells, right? Very nicely visible layer of cells, and that's the epithelium part. So covering layer of tissue is the epithelium of this organ. The organ in this question is the ovary, by the way, in case you are curious. And then we've got the nervous tissue. Don't worry, this tissue is usually not nervous, although it can make you nervous. Nervous tissue is made up of neurons. Remember neurons from the last video? So neurons combine to form the nervous tissue. And they can form different kinds of organs. One of them is nerves, or the other is brain, and the third is spinal cord. So neurons help make the nervous tissue. And what they're involved really is they help in transmission and interpretation of nerve impulses. This will be again covered in its own chapter, but let me just quickly elaborate on this. For example, you're looking at something, right? So you have these receptors, these visual receptors in your eyes that allow you to see light. So when you see the light, it generates electrical impulses. Those electrical impulses are meant to be carried to the brain somehow. And they have to be carried by nerves. So nerves would be the organ that carry that electrical impulse to the brain. And to be able to do this, the nerves have to contain cells that are able to, or tissues, that are able to conduct information from one place to another. And those cells are called neurons, and groups of neurons would take that sensory information of electrical impulses to the brain to be interpreted. And again, brain would also be made up of nervous tissue. So all in all, combined, the nervous tissue makes up the nerves, the brain, and the spinal cord. And together, they kind of look something like this. Very, very scary, right? Very scary. Again, nope. I don't want to look at it. Let's skip this part. Very scary. Okay, so next tissue is connective tissue. Connective tissue is a type of tissue that is that in the body that supports, binds, or separates other tissues or organs. So, for example, you could have something like a bone here. Here's a bone, right? That's an organ, right? And then you've got a muscle here. So on top of that, maybe this is the bicep muscle. Now you want the bone to be connected to the bicep, of course, because you want the bones to move as you contract or relax the bicep. So to be able to connect the two, you need a connecting tissue here, and that is called connective tissue. Okay, connective tissue. There's many different types of connective tissue, and they make up many different structures, but an example of this is a tendon, for example, right? A tendon is what helps link a muscle to a bone. You could also have a ligament, and ligaments help link either cartilage to bone or bone to bone. Okay, most probably you will not be questioned about what a connective tissue is. Okay, so tissues include connective tissue, nervous tissue, right, epithelial tissue, glandular tissue, muscle tissue. These tissues, multiple tissues can combine to make different organs, right? For example, the heart. The heart would be made up of the muscle tissue, and it would also be made up of the epithelial tissue and connective tissue, right? 3 tissues would make the heart, for example. Stomach would also be made up of similar tissues. The stomach would contain glandular tissue to secrete the enzymes like the acid, hydrochloric acid. It would also contain muscle tissue to help in pushing the food along and churning it, right? Some connective tissue as well as epithelial tissue. So very quickly again, remember the digestive system from a while ago. Basically we had a person, and then they would swallow food, and it would go into a pipe called the esophagus. The esophagus leads the food to the stomach, and the stomach would digest the food into a liquid called chyme, and then you would have the small intestine, right? And then you would have a bunch of small intestines here, and then eventually it merges into this large intestine. So that's the large intestine. All of these are organs that are part of the digestive system. And again, each of these organs are made up of different tissues. Then we got the liver. Does anybody know where the liver is situated? In a person, if this is a person, right, the liver is present on the right upper abdomen, okay? Inside the right upper abdomen, so right here of the person. Right upper abdomen, just below the lung, the right lung. Now, the liver is the largest internal organ of the body, and it has a couple of functions which are covered later in its own part. But just for introduction, liver helps in detoxify toxins in the blood, and it helps in storage of glucose, right? And it also helps in breakdown of excess amino acids. So if you eat a lot of proteins, right, more than you need, then you will have excess amino acids, and you want to break them down. And if you want to break them down, you will have the liver do a process called deamination, breaking down the amino acids and making a waste product called urea. Number 3, we've got the small intestine, which we just looked at. So after the stomach, we've got the small intestine. Small intestine helps in digestion and absorption of food, and we've already looked at how it is adapted to absorb food. Number 3, we've got the large intestine. The large intestine is not involved in absorption of food. However, it is involved in absorption of water and mineral ions from the digestive food. Brain made up of, and so therefore large intestine would also help in making feces. Then we've got the brain. The brain is made up of nervous tissue. Basically what the brain does is that it receives information from throughout the body. It is receiving the sensory information, for example, anything that you see, anything that you smell, anything that you taste, anything that you feel, temperature, pain, vibrations. All of that is going into the brain, and it is receiving these impulses, and it is able to interpret or understand them and then eventually generate a response. A response could be, for example, if a football is coming towards your face, then the response would be that you probably raise your hand to block it, or you duck down. Another example of this would be that that you're listening to this lecture, right? And your brain is generating a response of storing what I'm saying in memory so that you remember it when you go to your exams. Again, we've got glands. Glands, we already looked at the glandular tissue. So examples of glands would be the pituitary gland, thyroid gland, adrenal gland, ovaries, testes, and pancreas. Each of them will be discussed in the chapter dedicated to hormonal coordination. Okay? All right, so each of these organs are going to be discussed in more detail in their own systems. The first system that we're going to be starting with is the digestive system. Apart from the digestive system, we also have all these different systems of the body, like the nervous system, which is made up of the brain and the spinal cord and the nerves. We have the cardiovascular system, which is also called the circulatory system. It is made up of the heart, the blood vessels, and stuff. Endocrine system, right, which contains the glands, like the pituitary gland, the thyroid gland, the adrenal gland, musculoskeletal system, and the urinary system, which helps produce the urine. So far what we have learned is that how cells contribute to bigger structures and so on. But what about cells themselves? What are they made up of? What molecules are they made of, right? What molecules do we absorb from our food that help make our cells and then help make our body? That's a very important discussion that we need to take before going into the digestive system. We need to understand what molecules we need to take in from the food to be able to, for which we need the digestive system, okay? So in this section, we focus on biological molecules, for example, sugars, right, which help remember. In mitochondria, sugars like glucose is used for aerobic respiration to give us energy. Proteins help, discussed in its own portion as well, but proteins help in growth. Fats, right, also used for energy, and nucleic acids like DNA are used for for making genetic material. So number 1, we've got the sugars, carbohydrates. You know, sugars are also called saccharides, so I'm just going to write that down here. Sugars are also called saccharides. So sugars or carbohydrates, as the name suggests, carbo stands for carbon and hydrate stands for water, right? Hydrated. So these are hydrated carbons. They contain, for every carbon molecule, they have one water molecule, at least for glucose. That's how it works. Remember glucose, right? Its formula you should be aware of is C6H12 and O6. So C6H12O6, it seems like for every carbon, it's got a molecule of water. So maybe 6 molecules of water here, right, in its structure. Well, that's what carbohydrates are. Carbohydrates are hydrated carbons, and these are made up of, therefore, only carbon, hydrogen, and oxygen. These are used for making energy. and other cellular structures. So the energy that they make usually in aerobic respiration is around 4 kilocalories per gram. You don't have to remember this number for the sake of AQA at least. So there's different kinds of sugars, right? Different types of carbohydrates, different types of saccharides. Sugars can exist as single molecules. So you would have just a single molecule. That's number one. So you could have single molecule sugars, right? Single molecule sugars, like glucose is a single molecule sugar. Or you could have something like fructose, which is a single molecule sugar. Or you could have galactose, and so on. Or you could have, so single molecule sugars are also called monosugars or monosaccharides, okay? Monosugars or monosaccharides. Then we've got double sugars. Double sugars would be made up of two single sugars combined together. So you could have glucose and fructose, which give you something called sucrose. Or you could have glucose and glucose, right? Which give you something called maltose. Or you could have glucose and galactose, which give you lactose. Now, because these are two molecules making a sugar molecule, we call them di for two sugars, right? Disugars or disaccharides. Now, here's the thing. Both mono and disaccharides, they belong to simple sugars. These are simple sugars, right? These are very small, simple molecules. So we call them simple sugars. And it is very important to make this distinction in AQA because we're going to be doing some food tests for which we'll be testing simple sugars differently than complex sugars. So simple sugars include monosaccharides, which include glucose, fructose, and galactose, and disaccharides, which includes sucrose, maltose, and lactose. Simple sugars. Now, if you have long chains of these molecules, then you can have something called polysaccharides, right? For example, over here, right, I make a very long chain, right, of glucose, maybe even make some branches here, right? So that's a very big molecule, and that is a polysaccharide, right? A polysaccharide, because it's got multiple sugars, polysugars, polysaccharides, and that's a complex sugar. So let's write this down, complex sugar. An example of complex sugar in plants is starch. For example, starch in plants. And in animals, it's glycogen. Also, remember from cell wall? Remember what cell wall was made up of in plants? Cell wall in plants is made up of cellulose. Cellulose is also a polysaccharide, okay? Cellulose in plants is also a polysaccharide. Remember we said that carbohydrates are used not only for making energy, but also for making cellular structures. So that's one of the cellular structures that they help make. Okay, so complex sugars are larger molecules made up of many simple sugars chemically combined together in long chains. And now examples of these include starch in plants, glycogen in humans, and cellulose. Starch is the storage form of glucose in plants, and glycogen, oh, it's written here, so starch is a storage form of glucose in plants, which means that when plants make excess glucose, they store it in the form of starch, for example, in potatoes. Now, something very important for you to remember, and I want to highlight this as well, is that starch is digested by an enzyme called amylase. Okay, it is digested by an enzyme called amylase, and that's very, very important. Please remember this. Starch is digested by an enzyme called amylase. Now, this needs a little bit of a background. Let me just quickly show you the structure of starch again. So remember, starch, right, is a polysaccharide, which means it's got these long chains of glucose molecules connected together, right? And it's even got branches, and that's going to be present in plants. So what you do is you bring an enzyme called amylase. So here's an amylase, and you bring it. And what you do is this amylase, what this does is that it breaks it down into disaccharides, right? Double sugars. So it's going to break it down into disaccharides, break it here, break it there, right? And you get all of these disaccharides. So once you're getting these disaccharides, which are made up of glucose molecules, right? Here's a disaccharide and here's a disaccharide. So that's what amylase does. It breaks down starch into disaccharides made up of glucose molecules. And remember the name of the disaccharide that contains two glucose molecules in a structure? That's called maltose. Okay, maltose. Now, if you want to break down maltose into single sugars so that you can absorb them, that's another enzyme that you need. The other enzyme is called maltase. Maltase. So that's very easy to remember, right? Maltose is broken down by maltase. You can also try to remember the starch enzyme by this. This straight chain of starch, that's called amylose. Okay, the straight chain is called amylose. This is out of AQA, but I'm just telling you to make the name of the enzyme make sense. So amylose is a straight chain. So amylase is the enzyme that digests starch, right, into maltose. And then maltose is digested by maltase into glucose, individual glucose molecules, right? Individual glucose molecules. Now, as a group, these enzymes that are breaking down the sugars or carbohydrates, these enzymes, like maltase and amylase, are called carbohydrases. Okay, carbohydrases. I'll give you some other examples of carbohydrases just for understanding purposes. Remember lactose, right? Lactose is made up of a glucose molecule combined with the galactose molecule. Now, the enzyme that breaks down this bond and breaks down lactose, this is lactose. It's a sugar that is present in milk. A lot of the people are lactose intolerant. You've probably heard of this, and maybe a lot of you are also lactose intolerant. So the enzyme that breaks down lactose is called lactase, right? Lactase. So very easy to remember these names. Sucrose, remember sucrose, which is made up of a glucose molecule and a fructose molecule. Sucrase would be the enzyme that breaks it down, okay? So very easy to remember these names. Now, as a group, these are called carbohydrases, and that's what you need to know for the sake of AQA. Carbohydrases, that's number one, that's the name number one, and number two name is amylase. These two you need to remember, okay? You don't need to remember the rest of them, but I've just told you. All right. So next up, we have glycogen. Glycogen is the storage form of glucose in animals, right? So in humans, for example. If you want to store excess glucose, we convert the excess sugar into glycogen that is stored in our muscles and liver mostly. So why are starch and glycogen such suitable storage molecules, right? Both starch and glycogen are suitable for storage of glucose because a couple of things. Number one, they can store many glucose molecules in a small compact structure, right? You see how many molecules are present in this structure? So very quickly and very easily, they are able to store a lot of glucose in a very small space. Number two, they can very easily be broken down back into glucose. And number three, they do not affect the cell concentration. Remember osmosis, right? Osmosis, if there's a cell and it becomes very, very concentrated on the inside as compared to the outside, then water is going to start moving in. However, when you convert the glucose into starch and glycogen, it prevents the cell's concentration from building up. Okay, they do not affect the cell concentration. So if it exists as glucose, that's going to make the inside of the cell very concentrated. But if you convert that glucose into starch and glycogen, you don't have this anymore. And so you don't have water moving into the cell anymore, okay? And the third is the cellulose, which is a strong fibrous material which is used to make plant cell walls. This is also made up of long chains of glucose molecules, so kind of like this. You've got these long chains, right? And so these together will make a strong fiber, and then a lot of those fibers will make up cellulose, which makes the strong cell wall, okay? So we've got simple sugars and we have got the complex sugars. Now, a very high yield topic for your exams is the tests for simple sugars. And I want you to remember these tests by heart. The test for simple sugars is Benedict's test, and usually on the exam, this question will come for 3 marks, not 2 marks, okay? For 3 marks, as compared to the test for complex sugars like starch. Starch test is going to come for 2 marks. Benedict's test comes comes for 3 marks because it's got an extra step. So how do we conduct the Benedict's test? Well, here's how to do this. Basically, the blue colored, and you have to remember the color blue Benedict solution and the sugar solution are mixed in a test tube. Then it is heated in the water bath. That's the extra mark there. So you take the blue Benedict solution, they will give you 1 mark for mentioning the Benedict solution test, right? Mentioning the name Benedict solution, that's 1 mark. Then the test tube is heated in a water bath, that's 2 marks. So 1 mark for this, right? And 1 mark for this, and then the color change is observed. And then you mention the color change. If there's a color change from blue to red, right? Mentioning the color change from to, right? From which to which color. Then the tested solution is strongly positive for simple sugars. So that's the 3rd mark there. So 1 mark for mentioning Benedict solution, 1 mark for saying it is heated in a water bath, and 1 mark for saying that the color change is from here to here, right? If color change occurs, then the sample is positive, while if the color change does not occur, if it remains blue, then it is negative. The color change really, it goes from a spectrum of blue to red in the increasing order of concentration, right? So blue would be would mean no sugars because it's not changing. Becomes green, that's a little bit of sugar. Yellow is a medium amount of sugar, and then red would be the highest. Okay? All right, so I hope you guys don't mess around on this and always get full marks whenever you see the Benedict's test. I hope you guys feel happy because you know this. And next up we have starch test. So starch test question usually comes for 2 marks. This is because it's very simple, but basically what you do is you do the iodine test, right? The iodine solution is added, a few drops of orange iodine solution is added onto the sample that you're trying to test. And then the color change is for orange, from orange to blue-black. And that is 2 marks. So 1 mark for mentioning iodine solution test, right? Iodine solution test, 1 mark. And the 2nd mark is for telling the color change, and you get 2 marks for this. Okay, so an example is shown here in this photo. You have the orange iodine solution, which is poured onto a banana here that contains starch, and some potato here that contains starch, and look how it's turning blue-black. So again, on the right side you've got this orange solution, and on the left side you've got this blue-black solution, which is a positive test for complex sugar starch. So the next biological molecules in our course are lipids. Lipids, also called fats, right? You've probably heard of this. And in A levels, they're also referred to as triglycerides. Triglycerides and another lipid called cholesterol. I'm mentioning cholesterol here because we'll be talking a little bit about this in the heart disease chapter. Okay, so what are lipids? Lipids are insoluble molecules made up of carbon, hydrogen, and oxygen. These are insoluble, okay? And these are also made up of carbon, hydrogen, and oxygen. Now, these can be used for producing energy, and look at the energy that they produce, 9 kilocalories per gram. 9 kilocalories per gram. Remember the sugar's energy? The sugar's energy was 4 kilocalories per gram. So lipids actually produce 9 kilocalories per gram. So a question might arise in your mind as to why do we only use, or most of the time what we use is sugars for making energy. Why don't we switch to lipids, right? Because they give us a lot more energy than sugars do. So maybe you can look this up on the internet, and if I would very much appreciate it if you can code this question and see and answer what you find, okay? So anyways, let's continue. So can be used for producing energy, 9 kilocalories per gram, or for making cellular structures. For example, very importantly, the cell membrane. The cell membrane is made up of lipids. They can also be used for making steroid hormones. Steroid hormones include testosterone, which we'll do in its own chapter, estrogen, etc. So what is the structure of a fat molecule? A fat molecule kind of looks like this. So it looks like you have a molecule that is a glycerol molecule. Does anybody know what glycerol is? Does anyone know what alcohols are? Have you studied organic chemistry? If you studied organic chemistry, you might have studied something called functional groups, right? And alcohols and carboxylic acids. Alcohol functional group is that alcohols are molecules that have an OH, hydroxyl group, joined to them. A glycerol is a unique alcohol that has 3 hydroxyl groups. So one OH, another OH, another OH. 3 at the same time. At the same time, you will have 3 fatty acids that come in and try to bind to the glycerol molecule. So this is a glycerol. And you would have 3 fatty acids. Fatty acids are organic acids. These are carboxylic acids. Because these are carboxylic acids, they would have a carboxyl group. To some of you, this might be confusing. Hopefully, when you have done year 11 chemistry, then you can come back to this and review this. This is part of condensation polymerization in AQA GCSE chemistry, but also it is present in, if you're doing other boards chemistry, again, it is present in organic chemistry, which is usually done in year 11. So, we will have 3 fatty acid molecules. Fatty acid molecules would have long chains, and then at the very end, you would have a carboxyl group. Remember, a carboxyl group, carboxylic acid group, is C double bond O OH, right? That's what you learn in organic chemistry. C double bond O OH, right? Another fatty acid here, and then another fatty acid here. C double bond O OH. So, just like carboxylic acids, carboxylic acid groups and alcohols react in organic chemistry, same reaction will occur here. What will happen is you will have the H coming from this one and the OH coming from that one, making a molecule of water, right? And so when they form a molecule of water, now this oxygen can bind to something, and this carbon can bind to something, and so they end up forming a bond with each other, right? They have a bond with each other, and that's called an ester bond. And the reaction is called condensation reaction, because you make a molecule of water. Condensation reaction. Okay? So again, let me repeat this at the molecule below. So you have hydrogen that comes from the hydroxyl group, and OH that comes from the carboxylic acid group. And they make a molecule of water, and this is called condensation reaction. And because this occurs, now you have this oxygen that is able to form a bond with something, and this carbon that is able to form a bond with something, and they form an ester bond with each other. Okay, ester linkage. Then let's doing this for the 3rd time. We have the last fatty acid here. This is the fatty acid, and it's got the carboxyl group, and there's a hydroxyl group from the alcohol, the glycerol molecule, forms a molecule of water and forms an ester linkage with each other. So that is a triglyceride molecule. That is a fat molecule, right? That's a lipid molecule. So fat molecule consists of 1 glycerol and 3 fatty acids, right? 1, 2, and 3. We got 3 fatty acids and 1 glycerol molecule. Again, let me just quickly show you. This is a glycerol, and then we've got these 3 fatty acids that are attached to a glycerol molecule, and that's a fat molecule. Now, if you want to break this bond that is formed due to condensation reaction, during the formation of the bond, water was released, right? Water was released. If you want to break this bond, you got to put the water back in, right? You take this water, put it back in. So you break this bond, you lyse this bond using water. And therefore, that reaction to lyse the bond using water is called hydrolysis. Hydrolysis will be the reaction that occurs to break this bond using water. Hydrolysis occurs with enzymes that end with ases. Like, remember maltase? Maltase would also be hydrolysis. Amylase is also hydrolysis. And so for lipids, you would have an ase enzyme, a hydrolysis enzyme that breaks the ester bond between glycerol molecules and fatty acid molecules, and those enzymes are called lipases. Okay, lipases. So again, let's read this through. Structure of lipids. Each molecule of fat is made up of 3 fatty acids joined with 1 glycerol molecule through condensation reactions, right? That's part of year 11 chemistry. Enzymes that break down lipids are called lipases, okay? All right, so moving on. What are some of the tests for lipids? What are some of the tests for lipids? Well, one of the tests is that we have Sudan 3 test. Basically, what it is that you have equal amounts of water in sample containing fats into a test tube, and then you add a few drops of Sudan 3 solution, which a red stain layer then is observed on the surface of the test tube. So basically what I mean by this is that you have a test tube, right? And in this test tube you put some water, right? And you put some fatty material here, right? And you mix it together. And after mixing it, what you do is you add some Sudan 3 solution. And once you add Sudan 3 solution, what you will observe is a red colored emulsion that appears at the surface. And that means that the test is positive. If you don't see any red colored density that is forming on the surface or anywhere inside, then that means that the sample that you have is negative in lipids, okay? Another very important test, this I think usually comes from for 2 marks, while another very important test is the lipid emulsion test. By the way, a safety precaution for the Sudan 3 test is that you want to avoid skin-to-skin contact. And number 2, Sudan is flammable. So you want to be careful about that as well. Number 2, we've got the lipid emulsion test. This again comes for 3 marks. Why does it come for 3 marks? Because it's got an extra step, okay? An extra step. So what do you do here? What you do is that you take a test tube in which ethanol and lipid sample are mixed together. Then the resulting mixture is added to a water containing test tube. So again, you have a substance, right, and ethanol, and you stir it, shake it a little bit, and then you mix it with water. When you add it with water, what you will observe is that you will have a white-colored, cloudy emulsion, milky-colored emulsion, multicolored precipitate that is formed, which indicates the presence of lipids. Okay? So 3 steps. Number 1, you have a test tube that has the substance and ethanol. That's 1 mark. Number 2, you add it to water. And number 3, you get this milky, white-colored precipitate that is formed. And you get 3 marks for this. So I really hope that you guys get full marks on both of these every time you see them, okay? And next up we have proteins. Okay, so the next type of biological molecule are proteins. Proteins are molecules that are made up of amino acids, right? Proteins are molecules that are made up of amino acids. And amino acids are made up of carbon, hydrogen, oxygen, and nitrogen. Which is a new thing because remember, carbohydrates, they were only made up of carbon, hydrogen, and oxygen. Lipids, they were only made up of carbon, hydrogen, and oxygen. Proteins also contain nitrogen. Therefore, plants, they use nitrate ions to make amino acids, okay? Nitrate ions to make amino acids, which help in making proteins. Later on in year 11 chemistry, you also learn about something called NPK fertilizers, and they contain a lot of nitrate ions and nitrogen in them, so that the plants are able to get the nitrogen that they need for growth. So anyways, proteins, they have 2 functions. Number 1, and most of the time what they do is proteins help form cellular and other body structures, okay? Cellular and body structures. So for example, muscles and tendons, hormones, enzymes, receptors, antibodies. Do you know what an antibody is? Does anybody know what an antibody is? Antibodies are proteins that are used for immunity. They help fight off pathogens. And we'll do the details of this later in its own chapter. They can also be used to make energy, right? So 2 things. Mostly they're used to make cellular structures, but they can also be used to make energy. And the energy that they generate is 4.5 kilocalories per gram, which is very similar to the energy that is generated by carbohydrates. Carbohydrate is 4, proteins is 4.5, and lipids was 9 kilocalories. So proteins in structure are long folded chains of amino acids. How do they look like? Let me just quickly show you. So basically what happens is, remember where in the cell proteins are made? In the cell, proteins are made in The ribosomes. The ribosomes, what they do is they link amino acids together. So here's an amino acid, and then here's another amino acid, and so on. And so they linked different amino acids together by bonds called peptide bonds. Okay? Peptide bonds. So different kinds of amino acids are linked together by peptide bonds to form this long chain of amino acids connected by peptide bonds, and that's called a polypeptide chain. Polypeptide chain. The polypeptide chain is the very initial, very simple structure of a protein. Polypeptide chains can further also undergo folding to help make a three-dimensional structure, or they can just stay as it is, right? They can just stay as a simple structure, or they can just go ahead and fold. So let me just quickly draw this as a line. Now this line, or this polypeptide chain, can fold onto itself, kind of like this, right? Forming a specific structure, a specific 3D structure, if it wants to, or if it's supposed to. Or it can just stay like that, right? So this is a 3D structure, and that is a simple structure. Proteins can exist as either of them. Now, very importantly, you can heat the proteins. If you heat the proteins, this 3D structure will be lost because the three-dimensional structure is held by weak bonds. And so if you heat it, right, if you heat this, then what will happen is that the bonds that are holding the chain together are going to come off, and you're going to lose the three-dimensional structure that was there, okay? What you lose is the normal nature or normal natural shape of the protein. So when you lose the normal natural shape of the protein, we say that the protein is denatured, okay? Denatured. Not only is heat able to do this, which increases the kinetic energy of molecules so that the particles split apart and denature, but also pH, right? Extreme pHs, extreme acidity or alkalinity is also able to denature proteins. Now in total there are 20 amino acids, and these combine in multiple ways to make millions of proteins, okay? So there's a total of 20 amino acids in the world. Not more than this, but they make so many different proteins, right? Millions and billions of proteins. Proteins are digested by enzymes that break these bonds, the peptide bonds. So likewise, just like for lipids, we had enzymes called lipases, right? And for maltose, we had maltase, right? For carbohydrates, we had carbohydrases. Similarly, for proteins, we have proteases, and the ones that break peptide bonds, peptidases, right? Proteases, peptidases. An important protein is made by the stomach. It's called pepsin. Pepsin is a protein that is made by the stomach. So, how are amino acids linked together? This takes us to year 11 chemistry again. So if you're still not in year 11 yet, if you're not just reviewing it for your exams, then you can come back to this video when you study organic chemistry and review this. But this is for year 11 students. You learn something called condensation polymerization. And in that, you learn the structure of an amino acid. An amino acid structure, generally, remember there's 20 different kinds. Generally, an amino acid structure is that it has a central carbon. Then, on one side, that carbon is attached to a carboxylic acid group. Remember in organic chemistry, that's C11OOH, carboxylic acid group. And on the other side, it is attached to an amine group, NH2 group. Therefore, it is called an amino acid, right? Because NH2 is the amine group, and C11OOH is the carboxyl group. Remember, a carbon is present in group 4, so a carbon is able to form 4 bonds. 2 bonds, one with the carboxyl group, one with the amine group, then one with the hydrogen group, and one with the variable R group, right? R group. R group is a variable group that is different for each of the different 20 amino acids. So each of the 20 amino acids would have their own R groups, R1 to R20, and this could be anything. This could be a hydrogen, this could be CH3, this could be many, many different things, right? 20 different things for 20 different amino acids. So the general structure of an amino acid kind of looks like this. So how do 2 amino acids linked together in the ribosome? Well, for this, we'll have to draw another amino acid here. So let's get another amino acid here. So again, a central carbon, and then it would have an amine group on one side, right? And a carboxyl group on the other side. A hydrogen and a variable R group. So let's just call this R2 for the second amino acid, and let's call this R1 for the first amino acid. So how do these two link together in the ribosome? The answer to this is, again, condensation reaction. Condensation reaction. Remember condensation reaction? You get a hydrogen from one of the species and a hydroxyl group, OH, from the other one. So in this case, we have an OH here, and we get a hydrogen from this one, right? And they get married together, forming a molecule of water. Okay? And that is called a condensation reaction. Condensation reaction. So once this occurs, now this carbon is able to form something, a bond here, is able to link something here because the space has been freed. An electron has been freed and it can share the electron with something. Same goes for this nitrogen. Nitrogen is in group 5, it's already forming 1 and 2 bond here. The third, it will form with the carbon. And they form this bond, and this is called a peptide bond. And this would all occur in the ribosomes. And again, if you want to break this bond, then you got to put the water back in. Water is released when you make this bond. So if you want to break this bond, put water back in, and that will be called hydrolysis. And this will be done by enzymes like peptidases, which help break down proteins that we eat, proteases. For example, pepsin will help break down proteins that we eat. So a little bit of a reading, year 11 chemistry. This is just an extra addition to the biology course. You will not be tested about the details of this. This is just for a supplementary little section for students also taking chemistry for AQA and other boards. So amino acids, they contain a central carbon, which has 4 things attached to it: a hydrogen, an amine group, a carboxyl group, and a variable R group. 20 different amino acids would have 20 different R groups, and the hydroxyl from the carboxyl group and the hydrogen from the amine group, they combine together to form a molecule of water in a condensation reaction. This results in the formation of a peptide bond between adjacent amino acids. And again, if you want to break down this bond, break down the peptide bond, then you'll have to do hydrolysis on it using enzymes like peptidases and proteases. An example of a peptidase protease is pepsin. Okay, so a question for you guys. Where in the cell do you think these peptide bonds are formed? So I would love for you guys to quote your nicknames, this question, and your answer in the comments. And also answer all the other questions that I make during these videos, okay? All right, so next up you have tests for proteins. This is very simple and usually comes only for 2 marks. Right, it's not a 3-step question, so 2 marks only. Number 1 mark you will get for mentioning the correct solution. So which is the Biuret's solution, right? Add blue Biuret's solution to a test tube containing protein sample. Do not confuse this with Benedict's solution. Biuret's solution. So again, do not confuse a Biuret solution with a Benedict solution. Both solutions are blue, and Benedict's is used to test for simple sugars. Biuret's is used to test for proteins. This usually would come for 2 marks, and 1 mark would be for mentioning the word Biuret solution to test for proteins, and the other mark would be for mentioning the color change, which is from blue to purple or violet. Okay, so 1 mark for this, and 1 mark for this, and you get 2 marks. Hopefully this comes up on your exam and you're able to confidently do it. Okay, and that marks the end of this video. So hopefully I'll see you guys in the next one. We concluded a meme, so here's one. I'm on a seafood diet. This little boy, I see food and I eat it. A lot like me growing up. Alright, so if you guys are looking for extra help, if you want to study some other subjects, or you want to have exam guided practice, then you can reach out to us using the WhatsApp link that is provided below. And make sure to like and subscribe and share this beautiful channel with your friends, because it's free, right? It's free. So I'll see you guys in the next one, and B2 enzymes.